So I'd like to thank the Circulating Club and the Lasker Foundation for inviting me to give me this chance to talk about some of my work on circulating DNA. So this is a picture of myself taken over 30 years ago when I was still a medical student in Oxford. So as a medical student, we have to study different medical specialties. And one of those is obstetrics, where I learn about when a woman is pregnant, she would be very anxious to know about the health, including the genetic health of the baby. But to test the DNA or chromosomes from a baby, at that time, we would need to invasively sample some fetal materials, such as by this process of amniocentesis, when a big needle is inserted into the uterus and then some of the fluids is taken out. But every time we do that, there's a risk that we may harm or even kill the baby. So as a young medical student, so I was wondering, is it possible that we could do something safer? For example, by taking a blood sample from a mother and be able to test the genetic material from a baby. But this thinking, of course, at that time was against conventional belief, because we all know that our blood circulation system and our mother's blood circulation system are separate. So theoretically, it would be very difficult to do what I have imagined. But however, as a young medical student, I was thinking that maybe this conventional thinking is not completely correct. Maybe there's some sort of passaging of cells from the baby into the mother's blood. And so I was thinking, well, how can we prove that? So I was wondering, if the baby is a boy, then maybe there will be male cells that would enter in the mother's circulation, which will allow us to test them. So I persuade a number of professors in Oxford to allow me to actually test this in the laboratory. And one of the experiments, we use a probe which will bind to a Y chromosome. So every time it sticks to a Y chromosome cell, then you'll see a black dot like this. So here in this slide, you can see a number of male cells in the circulation of a pregnant woman. And also at that time, PCR was just coming in to play, the polymerase chain reaction. So we also tried to use a PCR for such a demonstration. And we were able to demonstrate there's some male fetal cells indeed. And this was published just as I was graduating to become a doctor. And then a few months later, Lancet actually did an editorial on this called "Is it a boy?" Because theoretically, you could use this technology to determine the sex of the baby. And then I then practiced as an intern after my graduation. But during that year, I was constantly thinking about this research because I thought that I've started it, and so I have some responsibility to finish it. So I've decided after my 1-year internship, I would actually return to Oxford and to do this full time as a PhD student. So it's a picture of myself as a PhD student in Oxford, where I can see that I'm completely wrapped up. The reason is because I'm using a Y chromosome as a marker. And of course, every cell in my body have a Y chromosome. So basically, I'm trying to protect my samples from contamination from myself. But the problem is that once I started working on this project full time, I realized it's not as simple as I had thought. Well, in theory, can demonstrate those cells exist. But however, they're very low concentrations such that I just could not get a reliable and robust way to actually do it day in, day out. So after 3.5 years of work, my fellowship ran out. And so the work wasn't going particularly well, but I was fortunate enough at that time that I met my wife, Alice, who was also doing a PhD in Oxford at that time. So the two of us graduated together in the summer of 1994. And then I went back into clinical practice and then do research also in my spare time. And then a few years has passed and 1997 came. So that was a year when actually Hong Kong went back to the sovereignty of China. So my wife and I thought that is a good time to go back because both of our family actually live in Hong Kong. So I joined the Chinese University of Hong Kong. You can see this is have a rather lovely campus in the northeastern part of Hong Kong. And then I have a chance to actually think back over the previous 8 years. What have I done wrong that I couldn't crack this problem? And interestingly, about 3 months before I left the UK for Hong Kong, I saw in this issue of Nature Medicine, there were 2 papers, which says that in patients with cancer, you can see cell-free cancer DNA in the plasma and serum of those cancer patients. And then suddenly, I had an interesting thought, because I thought the baby growing inside pregnant mother has some similarities to a tumor growing in the patients. Like, for example, the placenta is rather invasive, it implanted into uterus. So I was wondering whether the phenomenon, the cell-free phenomenon in cancer, would also equally apply to the fetal situation. So in other words, for the previous 8 years, I was trying to hunt for fetal cells in the mother's blood, but maybe I've been hunting in the wrong place. What I should be looking for is to look at the regions between the cells, which is occupied by this fluid called plasma. But at that time, I don't have a lot of resources. I've never handled plasma before. So I was wondering, how can I extract DNA from plasma? And then I recall when I was in Oxford in the college, I never quite enjoyed the college food. So every now and then I actually had enough and I might just go back to my room and cook some instant noodles. And of course, to cook instant noodles, you just boil some water and then put the noodles in, and a few minutes later, you have your meal. So I was wondering, can I just boil some of the plasma and serum and then maybe take a few drops of that soup to be tested by PCR? So imagine this is rather crude, but on the other hand, at that time, my resources were very limited, so this is all I could do. But interestingly, you can see that even with this very crude preparation, I was able to get a Y chromosome signal in some pregnant women and no signals in others. And then we find that the women with the positive signal all have baby boys, the one without all have baby girls. So it's the first demonstration of presence of fetal DNA in maternal plasma and serum. So it hadn't been demonstrated before because previously people just focus on cells and just throw away the plasma. And then we know that this cell-free DNA then is present very early on, from the 7 weeks of pregnancy onwards. And surprisingly, from the 10 weeks of pregnancy onwards, the concentration reaches 15% or above. And this is surprisingly high because imagine the very big difference in size between the baby, very small, and the mother, very large. And furthermore, we find that after delivery of baby, this fetal DNA is gone within 2 hours. So that was good news because it means that fetal DNA cannot persist from one pregnancy into the next. And this is important because if it could, then a woman who had pregnancy before might have fetal DNA from previous pregnancy, which would interfere with the accuracy of prenatal tests done in subsequent pregnancy. But we didn't see that. And then we start to actually test some prenatal diagnostic applications of this cell-free DNA technology. So first we start with the easy one. For example, we can determine the sex of the baby, which is useful because certain genetic diseases are sex-linked and which are predominantly affecting boys, like hemophilia and like Duchenne muscular dystrophy. And then we also show that you can use this technology to detect the blood group type of the baby, because occasionally, if the mother and baby have different blood group type, like if the mother's rhesus negative and the baby's rhesus positive, the mother could attack the baby by antibodies. So we show that for both sex determination and rhesus blood group typing, our technology is pretty accurate. And so within a few years, this technology was used in many laboratories around the world. And then we became more ambitious. So the number one reason why pregnant women go for prenatal testing is because they're worried the baby might have chromosome disorders like Down syndrome. Now, so Down syndrome is caused by the baby having an abnormal number of chromosomes. So in the healthy individuals, we have 23 pairs of chromosome, or 46. So in somebody with Down syndrome or trisomy 21, the number 21st pair, chromosome 21, instead of 2, he or she will have 3. And normally we detect Down syndrome by taking a cell from a tested individual and asking how many chromosome 21 there are within the cell. So you can see, for example, in this cell, there are 3 copies of chromosome 21. But the problem is that in our field, in the mother circulation, the DNA is in cell-free form. We don't have this cell membrane surrounding the DNA. And so it's impossible to use those conventional chromosome counting methods. So for about 10 years, we tried different methods. We tried to enrich for fetal DNA. And then we tried to use different ways. I mean, they work in some extent, but just not work well enough. So around 2007, then we actually decided to actually go back and develop a new type of approach. So this new type of approach will involve the ability to detect the Down syndrome without separating fetal and maternal DNA. And I'll give you an analogy. Let's say that you have a wallet that you can put in different number of coins, depends on which day of the week it is. So if I want to know how many coins you have in your wallet, one way to do that is ask you to show me your wallet and then count it. So this is like invasive prenatal diagnosis. But another way to do that, if I have a very accurate balance, I can ask you to stand on the balance with the wallet in your pocket. And then on a day when you have more coins in your wallet, you weight a little bit heavier. So provided my balance is precise enough. So basically what we have to do is to build a molecular balance to allow us to measure that little bit of difference in chromosome dosage in the baby. And if you try to do it with conventional PCR, then you will never reach the precision needed. So what we're thinking is this: conventional PCR, you test basically everything together. So here you have a number of DNA molecules. The yellow one is a fetus and the red one is from the mother. But what we're doing now is not to test all the DNA together, but to test them in multiple reactions. And the number of reactions is chosen so that each reaction will either have one molecule or no molecule. So in this sort of situation, the fetal DNA will be analyzed separate from mother's DNA. And basically you can have any precision you want. And that just by using a sufficient number of those reactions. This is called digital testing or single molecule testing. So we then show that using this digital testing, we can indeed detect the fetal Down syndrome non-invasively. And this was published in 2007. But interestingly, within a few weeks of our publication, there's another publication from another group with the same finding. And surprisingly, actually, both papers were submitted on the same day. So we know we have competitions. And of course, we also further need to improve this technology. Because while this digital PCR method works, it doesn't work perfectly. And the reason, one of the reasons is because to do PCR, you have to design primers to bind to the area you want to amplify. So let's say I go for one gene on chromosome 21, and then one gene on another chromosome. But the problem is that in chromosome 21, there's over 200 genes. So if you focus on just one gene, you're basically throwing away the information for the other genes or even the intergenic region. So it's not very efficient. So we have to think of some ways in which I can maximize the use of information in blood. So I thought about this way of random sequencing. So in other words, if you can imagine, I can sequence millions of DNA molecules in the plasma of a pregnant woman. And because I know the human genome sequence, I can map each of those sequences back to the chromosome from it comes from. And then I can measure statistics of the distribution, the counting of different chromosome fragments in the blood. And interesting, when we did this, we find it is surprisingly accurate. For example, you can see that the green and red signals are pregnancy involving male and female Down syndrome babies. And you can see the signal is completely differentiable from the normal pregnancies, which is those in blue and orange. So in the initial series of 28 cases, 14 with Down syndrome, 14 normal, with 100% accuracy. So we published this in 2008. And once again, within a few weeks of this publication, there's another publication from the same group on the same findings. So I become very curious about this group. And so I read up about the last author, Stephen Quick. So I searched on the Google and find that he's a professor in Stanford. But interestingly, I also find that he also graduated from Oxford. And also in '94, the year when I and my wife graduated. So remember, when I find that, I actually went back home and told my wife about this. And she very insightfully actually dig out this photo. And interesting, Steve Quick was actually here. So you see that somehow fate has brought us together. And also I find that a few years later, when I was elected into the US National Academy of Science, on the day when the announcement was made, of course, I saw my name and I was very delighted. And a few lines down, I saw Steve Quick's name. So This is a photograph taken at the NUS induction ceremony. So anyway, so now back to research. So we now have a prototype technology which seems to be very accurate in the ability to detect fetal Down syndrome. And so we have to do a large-scale clinical trial, which we spend the next 3 years doing, and actually find that to be very accurate. So the accuracy was above 99%. So we published this in 2011. So we think that this technology really can make a big benefit to prenatal testing. So we're eager to push it in the clinic. But of course, I couldn't do it just in the university. So I have to partner with some commercial companies. And of course, if you want to partner with commercial companies, then we have to have patents. So of course, for the last 25 years, we've been learning how to protect this intellectual property. But initially, of course, we're not very experienced. So I remember that the first company which our university licensed to is based in the UK. And initially, they sat on it and didn't do anything about it. So in the end, they thought that our IP wasn't very useful. So they just throw it back to us. And then I remember I went to a conference in Thailand and met this gentleman who is Dr. Charles Cantor, who at that time was the chief scientific officer of Circulating. So we started talking, and eventually our technology was licensed to Circulating. But in Stanford, of course, Steve has many experience with commercialization. So they licensed the technology to a company called Verinata, which was then acquired by Illumina. So unfortunately, whenever you have different companies have claim to the same sort of technology, they will start to fight lawsuit. And this happened for Circulating and Illumina. And so a number of lawsuits was fought. But luckily, eventually, they were settled. And so the Stanford and our university IP was pooled together. So anyway, so technology was actually launched clinically in the autumn of 2011. And to date, this technology is called non-invasive prenatal testing, or NIPT for short. And it was now performed in over 60 countries and was used by millions of pregnant women every year. So it seems that the initial goal that I had when I was a medical student had largely been fulfilled. And I was thinking, what else can I do beyond Down syndrome? Now, if you're trying to push this to the limit, then I will ask, well, can I sequence the whole genome of the baby by using this technology? But this is a hard nut to crack. The reason is because the human genome in the circulation is basically fragmented in millions of pieces. And then the fetal DNA is minority and is mixed in with the mother's DNA. So initially, I have no idea how do we do it. Until actually one day, I went to see this movie with my wife. And I remember the first 10 minutes of this movie is in 3D. So I remember I was sitting in the cinema and put on my 3D glasses and waiting for a movie to start. And then I saw the Harry Potter title fly towards me. And then my eye was sort of caught to this egg chair in 3D, which interestingly actually looks to me like a pair of chromosomes. And then suddenly my eye clicked. And I thought for every pair of chromosomes the baby has got, it has one copy from father and one copy from mother. So maybe I need to have 2 algorithms to solve this problem. So what I mean is this. So imagine, let's say this picture is a father's genome, and this is the mother's genome. So the baby will have one half from father and the other half from the mother. And now in the mother's blood, there's a lot of the mother's genome floating around, and we're trying to decipher the fetal genome, which is in yellow here. So remember, there are 2 algorithms. So first, we focus on the father's side first. So what I do is that I look at the father's genome and the mother's genome and try to find things which is only present on the father's side, but absent on the mother's side, but which is easy to find, like the flower. And then I go into the mother's blood and then try to hunt for the flower. So every time I find a flower, there's a little bit of fetal genome that it has inherited from its father. And then I zip all these flowers together, and out come the paternal half of fetal genome. Well, how about the mother's half? The mother's half is more difficult because the baby's DNA is swimming in this ocean of the mother's DNA. So any flower that the mothers give the baby, the mother will have it herself. So I couldn't use the previous approach. So I have to use something different. So I was thinking, well, in the mouse genome, the left-hand half and the right-hand half should normally be present in the ratio 1:1, just like my left hand and my right hand. But however, if the mother has passed on the right-hand half to the baby, and then baby is growing inside mother and release a little bit of that right-hand half back in the mother's DNA. So quantitatively, the right-hand half should be present at a high concentration than the left-hand half. So basically what you do is that go into the mother blood to count those halves. So as a matter of fact, each of those halves is a composite of different genetic polymorphic markers. So we call it a haplotype. So basically this approach I call relative haplotype dosage analysis, or RIDDLE. So basically what you go is that you go in the mother's blood and you count the number of halves. So the half which is in excess is a half inherited by the baby. And then you do it millions of times over. So to see if this general strategy would work or not, we recruited a couple from our hospital. And both of the couple are carriers of beta thalassemia, which is a genetic form of anemia, which is very common in China. And the mother is 12 weeks pregnant. And they take blood from her and then do a sequencing. Now, if you do Down syndrome screening, then you sequence several millions read and the test is 99% accurate. But for this project, we actually had to sequence 4 billion DNA molecules. So at the time when we did it was actually pretty expensive. It cost about $200,000 for this one case. And of course, now with a new sequence that will be cheaper. But eventually, this is the result. So this is a circles plot in which the signal is plotted in a circle like this. The chromosome you can see. And the blue signal is a fetal signal. So you can see basically an entire fetal genome is represented in the mother's plasma. And then we can focus on any region. For example, chromosome 11 is where the gene causing beta thalassemia is located, the beta globin gene. So you can zoom in and we find that the baby is a carrier of beta thalassemia. So basically by using this technology, you can basically do any prenatal testing on any genetic disease. And so this work was published in 2010. It was chosen to be on the cover of this journal. So it seems that basically the discovery of self-revealed DNA has really revolutionized prenatal testing. And you can also see how the original discovery was inspired by work done in cancer. So we thought that since the prenatal work is now so accurate, maybe it's time to actually feed back what we have learned back into the cancer field. So we start with a number of cancers. So one of the conditions we look at is this cancer of the head neck region, called nasopharyngeal cancer, or NPC for short. So it's a cancer which affects this part of the anatomy just behind the nose. And this cancer is particularly common in the southern part of China where I came from. Now, for example, a Cantonese man like myself, we have a lifetime risk of this cancer about 1 in 39. Another reason why we choose this cancer to study is because for this blood-based system to work, and we need to have some sort of genetic marker which is specifically associated in cancer, just like when I look at the Y chromosome as a marker for male fetal DNA. For this cancer, I need to find such a marker. But fortunately, for nasopharyngeal cancer, we have that marker in the form of Epstein-Barr virus, or EBV, because you can find EBV genomes in the nucleus of those tumor cells. And I was wondering whether in the course of this biology, an NPC tumor cell would release this EBV DNA into the bloodstream. And I still remember when I first proposed this project to some of my clinician colleagues, they don't believe that this will work because they think that most of us are chronic carriers of EBV. So this test is going to be very non-specific. But interesting, when we actually did experiment, I find that it's pretty good. So on the left-hand side here, you can see in the plasma of NPC patients, it really stuffed full of this EBV DNA. On the other hand, right-hand side, if the individual didn't have this cancer, then mostly it's negative. So we thought, well, it might be useful for this to be developed into an early detection test. Now, we think it's worth doing because if you look at this survival graph, if you detect this cancer in stage 1, like many other cancers, then actually the survival is very good. The long-term survival is over 90%. But on the other hand, discovered in stage 4, which is this purple line, then nearly 40% individual actually dies. Unfortunately, in Hong Kong, where we have a pretty good public healthcare system, 76% of this cancer only discovered in stage 3 and stage 4, so discovering late, and many people are dying. So we think that we should do something about it. So a few years ago, we launched a study in which we want to study 20,000 subjects over 3 years. We like to focus on the highest risk group, which are males between age 40 to 60. We want to see whether this blood-based detection system would allow us to pick up this cancer earlier than is otherwise possible. So every week we send a team out to different regions in Hong Kong, and every week we recruited about 200 subjects. In the end, we just recruited over 20,000 subjects, and through the screening, we could find 34 cases of this tumor. Now, this number is very informative because the incidence of this tumor in Hong Kong is about 35 over 100,000. And therefore, if you look at 20,000 people, statistically, you expect only 7 cases of this tumor. But somehow, we're getting 34 cases. So interpretation is that we're actually getting tumor which will present themselves over the next 4 to 5 years. So in other words, you should get them earlier than is normally possible. Now, for example, if we don't do the screening, as I mentioned to you just now, then most of the tumors detect in stage 3 and stage 4, which is an orange and red signal. But now with screening, we find that most of the cases is detected in stage 1 and stage 2, which is a green and blue signal. And also because we're getting them so early, we find that the survival is very good. So of the 34 cases, only 1 individual actually died of this disease. The rest are basically cured. And then you can see in the bottom here, if you don't use the screening, then the chance of them dying is much increased. As a matter of fact, it's increased 10 times. So this work was published in 2017 in the New England Journal of Medicine. In that year, the journal actually chose it to be one of their 10 notable articles that year. So we have now actually launched this test in Hong Kong, and we're going to actually hope to also launch it throughout the whole Guangdong region. But however, nasopharyngeal cancer is only first step. So we believe actually this circulating DNA approach can be equally applied to other cancers. But of course, one question is the entire tumor genome present in plasma, just like the fetal? So we actually use a very similar approach to what we did for Down syndrome to sequence the entire tumor genome. And we call this cancer genome scanning in plasma. And we find that basically can detect copy number aberrations, so amplification, deletion. And this is basically similar to what you detect the trisomy, because trisomy is almost like an amplification. You can see point mutations. And also in an individual with multiple tumors, you can see the mixture of those tumor DNA in the circulation. And then we wonder, okay, so if we can do that, maybe we can develop a generic test to screen for multiple types of cancer. But of course, to do that is more challenging because if you have somebody already known with cancer, then you can say this individual have some mutation like RAS mutation or P53 mutation. But if you have an individual who is thought that him or her to be healthy, you don't have that prior knowledge. So we need to have a class of a generic tumor marker which can be used across different tumors and across different individuals. So we decided to actually move from genetic to epigenetic, which is the modifications of the genome based on the same sequence. And one of the best known epigenetic marker is DNA methylation. So we actually developed this genome-wide methylation concept to screen for this cancer. We find that it works. So you can see this signal, the ring signal is the epigenetic signature. You can see basically one type of markers can be applied across multiple cancers. And this was published in 2013. And then assuming that you can detect, say, this individual has cancer. Of course, the next question that an individual or the doctor is going to ask us is, where is the cancer? But fortunately, epigenetics can help as well, because say within my bodies, the billions of cells, they have same genetic sequence, but they are modified epigenetically differently, depends on which part of bodies come from. So we can develop a technology where basically I do a methylation aware sequencing in the blood, and then compare the signal to methylation profiles from different organs in the body, and then can work out basically that the contribution from different organs. And you can imagine this technology can be applied to test for different conditions, different disease conditions, including cancer. So we call this technology plasma DNA tissue mapping. And then this combination technology was actually highlighted in this issue of MIT Tech Review as one of the 10 breakthroughs in 2015 as cancer liquid biopsies. Now, so if we think that this set of technology is going to have impact clinically if we're able to detect cancer earlier so that we can do something about that. So we think that we're eager for it to go to clinic as quickly as possible. So based on our experience with NIPT, we thought, well, one way to do that is to try to open our own company. So together with two of my colleagues, Rosa Chiu and Alan Chen, we founded a company called Serena, which basically stands for Circulating Nucleic Acid. Basically, we try to detect cancer at the early stage and to empower us to do something. But interestingly, at the same time, in the Silicon Valley, there's a company founded by Illumina called GRAIL. And see the mission statement is basically the same thing, to detect cancer early when it could be cured. So basically you can see that Serena and GRAIL basically have the same mission. And from our previous NIPT experience, then if you have that, then sooner or later you maybe have some sort of lawsuit, which is counterproductive. So around 2017, the two companies start to talk with each other, and eventually we decided to merge together. And I became a scientific co-founder of GRAIL. And this picture was taken with the first CEO of GRAIL, Jeff Huber, just to celebrate our merger. And this was very interesting. It was in the 2017 NBA finals. And so for the last few years, we've been working towards this multi-cancer screening test, which was actually launched in 2021, called a Gallery test, which is a multi-cancer early detection test. And of course, the field now is very hot. And there are multiple other companies and technologies. For example, this company called Thrive, which is based on technology developed in Johns Hopkins called CancerSeek. So hopefully this sort of technology will allow us to detect cancer earlier and eventually to cut down the mortality from this terrible disease. So next, I would like to spend the last part of my talk just to talk about some newer work with some rather unconventional technologies. So you remember that when we've talked about this first Down syndrome work, it's about counting DNA molecules. So a few years ago, I challenged some of my students to come up with an alternative solution to that problem. So can we develop another type of Down syndrome test, which is not based on counting? So we thought, well, apart from numbers, then we need to have some sort of characteristics of the circulating DNA which we can use. And we thought one of them, very fundamental characteristics, is the size of the DNA molecules. So if you measure the size of DNA molecules in plasma, this is what you get. So x-axis is the size from 50 to 200 base pair. And then you can see the fetal DNA in mother's blood is in blue, and the total DNA, which is made from mother, is in red. So basically, fetal DNA is a little bit shorter than mother's DNA. So we have this situation. So fetal DNA is short, and the mother's DNA is long. So it means the more fetal DNA you have in the blood, the shorter it would be. So for example, let's say that you have 2 tubes of maternal blood, one with 20% fetal DNA and one with 10%. And then you plot the size distribution. So when you look at the smaller size, the one with 20% will, of course, have the high peaks than the one with 10%. But on the other hand, if you look at the longer molecules, then of course the trend is reverse. But of course, if we try to give this graph to a clinician to look at, that will be very complicated. So we decide to actually encapsulate that essence into a metric, into a number, which we call size ratio, which is the ratio of short to long. And interestingly, you can see that if I plot the size ratio on the y-axis here, it's actually proportional to how much fetal DNA we have in the plasma. And this fetal DNA concentration is the #1 determinant of the accuracy of non-invasive prenatal testing. But previously, it was not that easy to calculate, because imagine I can do that based on a Y chromosome if the baby is a boy. If the baby is a girl, then it's more difficult. But now, with the use of size ratio, I can basically do it for all pregnancies. And then we wonder, can I push the size ratio even further? Now, so you can imagine we have some maternal DNA which is long, and some fetal DNA which is short. And normally they mix in to give you that size distribution. So now imagine the fetus have an extra copy of chromosome 21. And this extra copy of chromosome 21 is also releasing a short DNA into the mother's blood. And so it will theoretically drag down the size distribution of chromosome 21 in the mother's blood. So does it work this way? So we look at chromosome 21, and indeed, pregnancy involving trisomy 21 baby will have a different size distribution to the normal pregnancy and also pregnancy with other trisomies. You look at chromosome 18, then chromosome 18 size distribution is different. You look at chromosome 13, then the trisomy 13 pregnancy are different. So basically, by using this technology I call size-based molecular diagnostics, we can basically, is another way to detect fetal trisomies. So now on the market, there are products which incorporate the counting plus the sizing together so that you can have a more accurate result with fewer sequencing reads. But this size is only the beginning of a new type of markers, which is based on the fragmentation of DNA. So this fragmentation is because of enzymes called nucleases. And actually this whole field now has a name called fragmentomics. So size is one of those. And it's also like, almost like we go to a barber for a haircut. Depends on what of diseases the barber use will govern the type of cut we have. It could be a jagged cut or very clean cut. So similarly, in this situation, depends on what sort of nuclease is used, then actually the DNA could be jagged ends or blunt ends, or even some nuclease of preference of particular sequences of so-called end motifs. So this whole field of fragmentomics will give us many new markers for prenatal testing and for cancer and other disorders. So now we'll talk about implication of this fragmentation. So now I'd like to look at the opposite direction, is about how intact it would be. And the reason why I'm thinking about this is this. I showed you this graph just now. And of course, one question is that this graph is generated by some sort of sequencer. It's actually generated by Illumina sequencer. And of course, that type of sequencer is a short read sequencer. So we're wondering whether the type of sequencer you use might somehow bias to your observation. It's almost like if I wear spectacles of different colored glasses, then when I look in the world, of course, the world will look different color. So I was wondering, okay, let's say if I change the Illumina short read sequencer to one of the newer long read sequencer, single molecule sequencer, such as that from Pacific Bioscience, what would happen? And interesting, this is what happens. So for example, you use a short read sequencer, then of course, very few molecules are over 200 base pair. But now if I use a single molecule long read sequencer, then suddenly I see a whole populations of long cell-free DNA, which was previously invisible. And they're actually quite substantial because if you look at the first trimester of pregnancy, a median of 15% of those DNA are long. And second trimester go up to 20%, and third trimester go up to 30%. And also because the DNA is so long, then theoretically should contain more information. And one of those is genetic information, like the polymorphism we mentioned. Because you look at single nucleotide polymorphisms SNP, on average, every 1,000 base pair, there may be one SNP. And previously, if you look at very short molecules like 180 base pair, then you'll need several molecules before you encounter one SNP statistically. But now, by looking at this super long cell-free DNA, like this one, a fetal DNA of 16 kilobase, one molecule, we have multiple SNPs in there. So in other words, I can construct a haplotype just on one molecule. And this is another molecule from mother, 24 kilobase. Once again, one molecule, a whole haplotype. And also because it's so long, that apart from genetic information, you also carry epigenetic information. And of course, I told you previously about this technology called plasma DNA tissue mapping that we've developed. But this technology at that time will require a methylation-aware sequencing, which is based on treatment with a chemical called bisulfite. But bisulfite is quite a nasty chemical because in some publications said it can kill maybe 90% of DNA put in. And we're also very worried that the bisulfite might even make some fragment the long molecules which are already there into shorter one. So we decided to develop a technology which is maybe bisulfite free. So we're wondering, when I do the single molecule sequencing, when the polymerase is copying the DNA, is it possible that somehow the signal involving epigenetics is already incorporated into it? And previously, Pacific Bioscience have tried to look into this, but at that time, they find that accuracy is only like 5%, so it's not practical. So we wonder if I look at it holistically, look at the sequence context, look at the duration of the signal, and about the gaps, the time gap between different signals, can I deduce it? It's almost like when you're driving, you see an obstacle. Before you come to an obstacle, you slow down your car, and then you drive around the obstacle, and then you speed again. So now it's just, even without looking at the obstacle myself, just by looking at your trajectory and speed, etc., I can deduce there's an obstacle there. So this is the concept. So we developed this concept called the holistic kinetic model, HK model. So for example, you want to look into this methylation status of this CpG site, you can actually look at the sequence context, and you can measure those different kinetic parameters in that region and put in the table like this. And because the DNA is double-stranded, you sequence, you got this on one strand, and you got that on the other strand, and you combine the two tables together. And then to me, this table is a two-dimensional structure. So maybe we can actually use some of the artificial intelligence techniques which are developed to analyze pictures, to analyze the tables, such as the convolutional neural network, CNN. So we train such a network, and interestingly, we find it's able to actually generate or deduce the methylation status. And the methylation status actually correlate very well with a gold standard, which is by bisulfite sequencing. Now, for example, we can see that we look at blood cells, we look at placenta, we look at tumor cell line. It's once again a circles plot. The genome is plotted this way. And inner circle is the HK model, and outer ring is the bisulfite sequencing. And the color is basically how much methylation we have. You can see that the correlation is very good. And correlation coefficient range from 0.85 all the way to 0.98. And so this work was published early last year. So we find that by using this HK model and this sequencing, we're able to detect those long cell-free DNA in blood, which because they're so long, they have multiple CpG sites. And just on the CpG signal, I can deduce where it comes from. And the accuracy is pretty high, it's 0.91. So if it's 1.0, it's perfect. So 0.91 is pretty high. And then because it's so good, then we think maybe I can also use it for non-invasive prenatal testing of single gene diseases, which, as I mentioned to you just now, the previous gold standard is this technology called relative haplotype dosage analysis, which is accurate, but it requires many, many reads to reach saturation. But now by using those long reads, I can see that we can reach saturation much earlier. And this work was just published over Christmas last year. So now, of course, now we've discovered long cell-free DNA in pregnancy. Now, of course, the next question is, can I see that in cancer? And the answer is yes. So once again, in cancer patient, you can also see all those previously undiscovered super long cell-free DNA. And those super long cell-free DNA also contain the cancer-associated mutations and the cancer-associated epigenetic changes. Now, so we think that discovery this super long cell-free DNA is interesting. It's almost like for the last 25 years, when you're working with short molecules, you're based on the text message sent from fetus and the cancer to us. Each message only have 10 words or 20 words. But now with the super long cell-free DNA, for the first time, we can see the whole word documents from a fetus or from a tumor. So we believe that actually this long cell-free DNA diagnostics and direct methylation analysis will allow us to see details invisible before. So there will be many interesting diagnostic and research applications. So finally, in summary, hope I've convinced you that plasma DNA represent a treasure trove of molecular diagnostics. And throughout this work, it's useful to actually work on two phenomena in parallel, the fetus and the cancer. So seeing similarities in apparently different phenomena. In the fetal side, non-invasive prenatal testing is now already standard of care in the field. And the cancer liquid biopsy is coming in. And so I think this multi-cancer screening technology has a potential to eventually save lives. And we talk a little bit about the emerging field of fragmentomics. And also, and about this long cell-free DNA diagnostics. And finally, from my experience, I believe there's some synergy between academic research and commercialization. And I think it's useful for us academic scientists to also involve in the commercialization process so as to try to also influence the direction in which we think is best for the field. So finally, I'd like to thank individuals from my group to generating data which I present to you today. And thank you very much for your time.